Imagine a security system so advanced, it can take a mugshot of an intruder, distribute that photo to like trillions of guards, and then remember that exact face for 80 years.
Yeah, it's pretty wild when you put it like that.
Right. I mean, that is your immune system. But what happens when the intruder learns to wear an invisibility cloak or, figures out how to actually live inside the very guards designed to destroy them?
That's where things get really complicated.
Exactly. So welcome to the deep dive. If you are a medical student prepping for boards or a young professional stepping into the clinical world, or honestly just an insanely curious learner who is fascinated by human biology, you are definitely going to want to take notes today.
Oh, absolutely. Grab a pen.
We are looking at this incredibly dense, highly detailed stack of excerpts from the 2021 USMLE Step 1 lecture notes on immunology and microbiology. And our mission today is to basically translate these complex medical texts into clear, actionable knowledge about the body's immune defenses. And, of course, the medically relevant pathogens trying to bypass them.
Right, the bugs themselves.
Yeah. So, okay, let's unpack this.
Yeah.
Because the human body is, it's essentially the site of this unending microscopic war. And the stakes are literally life and death.
They really are. And for anyone listening who is, preparing for a clinical practice or board exams, let's establish a guiding principle right now. Rote memorization is a complete trap.
Oh, 100%.
Right. Simply flashcarding a list of bacteria and their lab values, well, that's not going to save your patient. And it definitely won't pass your boards.
No, it won't.
Understanding the why and the how behind the pathology.
The underlying mechanisms.
Exactly. The underlying physiological mechanisms of these battles. That is the absolute key to mastering infectious disease.
So if we want to understand how pathogens break in, I guess we first need to understand the security system itself, right? The texts describe the body as relying on this two-tiered defense grid.
Right, the innate immune system and the adaptive immune system.
Yeah, and they say the innate system is the first line of defense. So this is the security you were born with, right? Like present with or without any previous exposure to a bug.
Yes, intrinsically present at birth.
I always picture innate immunity like the bouncers at a busy nightclub. They're big, they act fast, and they just deal with obvious trouble immediately. But how do they actually know who to throw out?
Well, they operate using very broad rules. Those bouncers are looking for what the texts call PMPs, pathogen-associated molecular patterns.
PMPs, okay.
Think of PMPs as the biological equivalent of someone walking into a club carrying a baseball bat.
Right. You don't need to ask questions.
Exactly. The bouncer doesn't need to know the person's name or their backstory. The bat is just a universal sign of trouble. So the innate system uses anatomic barriers like your skin and physiologic barriers like stomach acid.
Just to burn them up.
Right. And cellular components like phagocytes to neutralize those really obvious threats. But that speed comes with a major limitation.
What?
Zero immunologic memory.
Wow. Right. So a bouncer will throw the exact same troublemaker out 3 nights in a row and never actually remember his face.
Yep. Every time is like the first time.
Which seems like a massive vulnerability, doesn't it? Like, if the innate system can't learn, how do we ever build long-term immunity?
Well, that vulnerability is exactly why the second tier evolved, the adaptive immune system. This system is composed of your B and T lymphocytes.
Okay, the specialized cells.
Right. While the innate bouncers act in minutes, the adaptive response takes about, 1 to 2 weeks to fully activate after a primary infection.
So it's much slower.
It is. It trades speed for extensive diversity and just incredible specificity. And most importantly, it generates that immunologic memory.
So if the innate system is the bouncers, the adaptive system is like a highly specialized detective task force.
That's a great way to look at it.
Yeah, like it takes them a week to assemble. But once they profile the specific culprit, they neutralize them, and they will forever remember that exact criminal's face.
Right.
If that bug ever shows up again in 10 years, the detectives are literally waiting for them on day 1. But wait, these two systems aren't just operating in silos, right? Like, how do the bouncers talk to the detectives?
So they operate in this really powerful positive feedback loop. It's called antigen presentation.
Antigen presentation.
Right. When innate phagocytes consume a pathogen, they don't just, digest it and move on. They act like informants.
Oh, I like that.
Yeah. They process the pathogen, extract a very specific protein.
The antigen.
Exactly, the antigen. And they physically present it to the T lymphocytes. That interaction stimulates the T cells to release cytokines, which are these chemical alarm signals that just trigger the massive, specialized adaptive immune response.
Okay, so let's get into the weapons of those adaptive detectives, because I was reading the notes on B cells and T cells, and they seem to operate very differently.
Oh, fundamentally differently.
Yeah. The text mentions that B cell receptors have this flexible hinge region and two combining sites, and they can actually be secreted into the blood as antibodies.
Right, they float freely.
But then it noted that IgM, which is a specific type of antibody, is secreted as a pentamer held together by a J chain. I got really hung up on this. How does a single cell secrete something that massive? I mean, it sounds like trying to push a whole car out of a mail slot.
It really does, but it's an incredible feat of cellular engineering. So a pentamer means it is literally 5 distinct antibody molecules linked together in a star shape.
5 of them.
Yep. The B cell synthesizes these individual pieces and uses that J chain, the joining chain, to clasp them all together right as they are being exported.
That's wild.
It is. It creates this massive 10-armed molecular net that is phenomenally good at clumping pathogens together in the bloodstream before they can spread.
Okay, so B cells can shoot these massive antibody nets out into the bloodstream. But then the text says T cell receptors are rigid, they only have one combining site, and they require something called CD3 for signal transduction. And they are never secreted.
Never secreted, right.
Wait, if T cells are the elite generals of the adaptive system, why are they stuck doing hand-to-hand combat? Why wouldn't they just evolve to shoot their receptors like B cells do? Isn't that, like, incredibly inefficient?
Well, if we connect this to the bigger picture, we see it's actually a vital safety mechanism.
Oh, really? Safety for us.
Exactly. T cells, particularly cytotoxic T cells, possess the ability to order cells to undergo apoptosis.
Which is programmed cell death.
Right. If T cells could just secrete their receptors and trigger cell death from a distance, the collateral damage to your own healthy tissue would be absolutely catastrophic.
Like a biological bomb going off?
Precisely. So by requiring physical cell-to-cell contact, the body ensures that this lethal force is highly controlled and targeted.
Oh, wow. Okay, that makes so much sense.
And that mention of CD3. Think of the T cell receptor as the keyhole that recognizes the pathogen, and CD3 is the ignition switch.
So you need both.
Yep. Without CD3 relaying the signal inside the cell, the T cell just won't fire.
That is fascinating. The body is deliberately restricting the adaptive system to prevent friendly fire. And the text also points out that the whole system must be strictly self-limiting, too. Like, once the infection is cleared, everything has to return to a baseline resting state just to conserve energy.
That's right. Because if it fails to self-limit, you are left with uncontrolled cellular proliferation.
Which is bad.
Very bad. Clinically, an immune system that forgets how to turn itself off often presents as leukemia or lymphoma.
Wow.
Furthermore, the system must be perfectly capable of distinguishing self from non-self. When that central tolerance breaks down, the detectives start profiling and attacking your own organs.
Autoimmune diseases.
Exactly. That's the root cause.
Which brings up a really important question. If we understand how this perfectly balanced system is supposed to work, what happens when it's broken from the start?
Immunodeficiency.
Right. The texts dive into primary immunodeficiencies, noting they are usually pediatric. But what really caught my eye is that these infants don't typically present with severe infections on day 1. It usually happens around, like, 6 months of age.
This is a critical timeline for any future clinician to recognize. You see, during pregnancy, maternal IgG antibodies cross the placenta and protect the fetus.
Oh, so they borrow mom's immunity.
Exactly. But antibodies don't last forever. They have a distinct half-life. By about 6 months of age, those borrowed maternal antibodies are nearly completely depleted in the infant's bloodstream.
So here's where it gets really interesting. If a baby with an immunodeficiency relies entirely on mom's IgG for those first 6 months, are their innate bouncers just totally offline during that time? Or is it only the adaptive detectives that are missing?
No, that's a great distinction. The infant's innate system, the phagocytes, the physical barriers, that is fully active from birth.
Okay.
But here is the mechanical flaw. Without maternal IgG to act as basically tags, the innate system is functionally blind to certain stealthy threats. Wait, blind.
Yeah. That tagging process is called opsonization. Think of antibodies like biological highlighters. The bouncers are at the door, but without the highlighters illuminating the microscopic intruders, the phagocytes physically cannot grab them.
That makes perfect sense. So let's talk about what happens when the bouncers themselves are genetically defective. The texts highlight a terrifying phagocytic defect called Chediak-Higashi syndrome.
Oh, yes. Chediak-Higashi.
It's caused by a nonsense mutation in the CHS1 or LYST protein, and it says this leads to aberrant fusion of vesicles. Now, for those of us who aren't, cell biologists, what does a vesicle fusion problem actually look like in a patient?
Well, to understand the pathology, picture the inside of a macrophage.
Yeah.
Normally, it engulfs a bacterium into a temporary stomach called a phagosome.
Okay, so it swallows the bug.
Right. Then a lysosome, which is a separate vesicle full of destructive acid and enzymes, must fuse with that phagosome to digest the bug.
Like pouring acid into the stomach. Yes. But in Chediak-Higashi, that fusion is broken. The macrophage swallows the bacteria, but the digestive enzymes are stuck in a completely different part of the cell.
Oh, no. So the bacteria is
It just sits there, alive.
Wow.
Clinically, you see severe recurrent infections because the bugs aren't dying. But uniquely, you also see partial albinism.
Wait. Albinism. How does an immune defect change skin and hair color? That doesn't seem related at all.
It's fascinating, right? It's because the exact same protein required to move lysosomes in immune cells is also required to transport melanin pigment in skin cells.
Are you kidding me?
No, the pigment is manufactured, but it just can't be delivered to the hair or skin. It's a fundamental cellular trafficking failure with multi-system consequences.
Wow. A single broken protein pathway breaks both immunity and pigmentation. That is crazy. Another defect mentioned is hyperimmunoglobulin E syndrome, or Job syndrome.
SIP syndrome, yes.
This one is a defect in the JAK-STAT signaling pathway, leading to impaired Th17 cell function. Again, breaking down the jargon for a second, what is JAK-STAT and why do we care so much about Th17 cells?
So the JAK-STAT pathway is essentially the cellular telephone wire. It transmits chemical signals from the outside of the cell directly to the DNA.
Okay, so it's communication.
Right. When that wire is cut, the cell literally can't respond to alarms. These 17 cells normally use that wire to recruit neutrophils, which are the heavy infantry of the innate system, to sites of infection.
Right.
Without C17 function, patients develop severe sinopulmonary infections and these classic cold staphylococcal abscesses.
Wait, cold abscesses?
Yes,
Yeah.
They are cold because without the neutrophils rushing in, you completely lack the normal heat, swelling, and redness of typical inflammation.
It's just a silent growing infection.
Exactly.
So that is the system underperforming. On the flip side, we have hypersensitivity, where the system violently overreacts.
Yeah.
The texts outline 4 types, but let's just look at the mechanics of the first 3 to start. Type I is immediate hypersensitivity. This is your classic, like peanut allergy or anaphylaxis, right? The text says it starts with Th2 cells, producing IgE antibodies that bind to mast cells.
And this is where the mechanism really dictates the clinical timeline. When an allergen binds to that IgE, the mast cell degranulates. It essentially explodes with chemicals.
Right, releasing histamine.
Yes. The immediate reaction, happening within minutes, is driven by preformed molecules like histamine, which causes your blood vessels to dilate and leak.
That's the fast part.
But there is a late-phase reaction hours later. This is driven by newly synthesized lipids called prostaglandins and leukotrienes, and those cause prolonged tissue damage and airway constriction.
Okay, so if type I is allergies, type 2 is antibody-mediated hypersensitivity. This is where your own IgM and IgG antibodies mistakenly bind directly to your own cell surfaces or extracellular matrix.
Right, it's direct, targeted, friendly fire.
But I always get type 2 mixed up with type 3, which is immune complex hypersensitivity. It sounds so similar.
Yeah.
What is the fundamental difference in how they actually cause damage?
The difference really lies in the target. In type 2, the antibody attacks a fixed structure, like an antigen permanently attached to a red blood cell.
Okay, fixed.
Yeah. But in type 3, the antibodies bind to soluble antigens. These are tiny proteins just floating freely in the blood.
Oh, I see.
They clump together into massive cross-linked webs called immune complexes. And these webs are too big to be cleared easily, so they just get stuck in the body's filtering systems.
So they just clog things up.
Exactly. Depending on where they settle, they recruit inflammatory cells and cause distinct syndromes. If they deposit in the joints, you get arthritis. If they deposit in the blood vessels, you get vasculitis. In the kidneys, nephritis.
Wow. Okay, so we've dissected the host defenses and how they malfunction. But if our adaptive immune system is so incredibly specific and remembers everything, how do we still get sick?
Pathogen evolution.
Right. Let's look at their armor and their stealth. But first, a quick note on the nomenclature, so we are all speaking the same clinical language. When you read the texts, genus and species names are always italicized, and bacterial family names always end in the suffix ACEAE.
Good to remember for the exams.
Definitely. So let's talk about physical armor first. Spore-forming bacteria. These bugs contain a substance called calcium dipicolinate. What did that actually do?
Calcium dipicolinate acts like a chemical dehydration shield. It pulls water out of the spore and stabilizes the bacterial DNA. This makes the spore incredibly resilient to boiling heat, extreme drying, and even harsh chemical disinfectants.
They are indestructible.
Pretty close. The main genera to memorize here are Bacillus and Clostridium. If they form a spore, they can survive dormant in the soil or, on a hospital bed rail for literally years until they find a welcoming host.
That is terrifying.
Yeah.
Then you have the stealth operators, intracellular pathogens. These bacteria don't just hide from the bouncers, they literally hide inside the host's cells.
Yeah, they use our cells as bunkers.
The texts classify them into two groups, facultative and obligate. Facultative means they have the capacity to live inside the very macrophages that are designed to kill them, which is just a wild evolutionary flex.
Truly.
Obligate pathogens, on the other hand, physically lack the machinery to replicate outside of host cells.
Exactly. And because they are hiding inside human cells, circulating antibodies can't reach them at all.
So how do we fight them?
They can only be fought off by cell-mediated immunity. Specifically, Th1 cells directing macrophages to aggressively digest their own contents, and CD8-positive cytotoxic T cells destroying the infected cells entirely.
Burn the house down to get the mug.
Basically, yeah.
But the ultimate evasion tool, the one that shows up in literally every major clinical vignette, is the microbial capsule. The text describes it as a polysaccharide layer. I always picture the capsule like trying to pick up a wet bar of soap.
Or a greased pig.
Right. The macrophage reaches out, but its receptors physically cannot get a grip on those slippery sugar molecules to pull the bacteria inside. They just slide right off.
And this is huge for the boards. The classic mnemonic for the heavy hitters is, some killers have pretty nice capsules.
Let's run through those. That stands for Streptococcus pneumonia, Klebsiella pneumonia, Haemophilus influenza, Pseudomonas aeruginosa.
Which notably produces a thick slime layer that is just devastating to cystic fibrosis patients.
Right. Then Neisseria meningitis, and finally, Cryptococcus neoformans.
Now, what's fascinating here is that almost every organism on that list is a bacterium.
Yeah, I noticed that.
But Cryptococcus neoformans is a yeast. It's a fungus.
Wait, why does a fungus have a bacterial invisibility cloak?
Well, this raises an important question about evolutionary adaptation. Cryptococcus developing a thick polysaccharide capsule is a massive biological anomaly.
Which makes it highly testable.
Oh, it's a phenomenal diagnostic differentiator on exams. Because of this capsule, it requires a robust, coordinated immune response to clear. This is why it specifically targets immunocompromised individuals, such as patients with untreated AIDS. Because AIDS depletes CD4-positive T cells, the immune system simply cannot activate the macrophages to deal with this heavily armored yeast.
And that leads to severe meningoencephalitis.
Yes.
Which bridges us perfectly into seeing these bugs in action. Understanding microbiology in a vacuum is kind of useless, right? Let's look at how meningitis presents differently based entirely on the age of the patient. Let's start with neonates.
In newborns, the major threat is Listeria monocytogenes. If you perform a lumbar puncture and the lab reports a gram-positive motile rod, you are absolutely looking at Listeria.
Good to know.
It's uniquely dangerous because it can cross the placenta or infect the infant during a vaginal delivery.
Okay, so moving up to unvaccinated children, roughly 3 months to 2 years old, where that maternal IgG has faded out, the classic culprit is Haemophilus influenza type B.
Also known as HIBE.
Right, under the microscope, this is a gram-negative pleomorphic rod. Now, I had to look up pleomorphic. It essentially means shape-shifting. It doesn't have a uniform structure, and it relies heavily on that slippery capsule we mentioned earlier to survive in the blood.
Then we shift to young adults. This one famously spreads in crowded environments like college dormitories.
Oh, dorm rooms.
Yeah. The pathogen here is Neisseria meningitis. It is a gram-negative diplococcus, meaning it looks like two berries stuck together.
Okay, 2 berries.
Clinically, you'll see a stiff neck, a rapid fever, and crucially, a petechial rash caused by microhemorrhages in the skin.
Now, here is the classic metabolic trick for distinguishing Neisseria species. Neisseria meningitis ferments maltose. M for meningitis, M for
Yep.
multiple.
Exactly. Its cousin, Neisseria gonorrhea, only ferments glucose.
That is such a clean diagnostic separator. I love that. Finally, in the elderly population, the leading cause is Streptococcus pneumonia. The lab will show a gram-positive coccus that is alpha-hemolytic, meaning if you grow it on a blood agar plate, it physically bursts the red blood cells, leaving this greenish halo. And it is sensitive to the chemical optogen.
Spot on. Now let's step away from the nervous system and look at the GI tract for a second. A standout in gastroenteritis is Campylobacter jejuni.
Oh yeah, the text said a lot on this one.
The lab clues are highly specific. It is a gram-negative curved rod with polar flagella, often appearing in pairs that look like a seagull under the microscope.
The seagull, right.
But here's the mechanism that really matters. It thrives at 42 degrees Celsius, which is about 107.6 degrees Fahrenheit.
And why 42 degrees Celsius?
Yeah.
Because that is the exact normal body temperature of a bird,
Yes.
which totally explains why Campylobacter is so commonly transmitted via undercooked poultry. It literally evolved to thrive in the avian gut.
Incredible adaptation.
One more respiratory vignette, atypical pneumonia. The lab says the organism is non-gram staining. Why? Because Mycoplasma pneumonia physically lacks a standard cell wall.
It's completely missing one.
And because of that structural deficit, it strictly requires cholesterol added to the laboratory media just to maintain its flimsy membrane.
Exactly.
So what does this all mean? When you are reading these dense lecture notes, learning that a bug grows at exactly 42 degrees, or demands cholesterol, or ferments maltose, I mean, it feels like you're just memorizing obscure trivia.
Which you aren't.
Right, you aren't. These physiological quirks are exact diagnostic cheat codes.
They allow you to rapidly rule out 50 other diseases and target your treatment to save the patient right in front of you. The lab values are just the biology speaking to you. You just have to know how to translate it.
Which brings us to the end of our deep dive. But before we go, as promised, let's lock in this knowledge with a quick clinical review exercise.
Oh, I like this. Let's test your synthesis.
Yeah.
So to the listener, a neonate presents to the emergency department with a high fever and signs of meningitis. You perform a lumbar puncture. The lab evaluates the cerebrospinal fluid and reveals an organism that is a gram-positive motile rod. Based on everything we've unpacked today, what is the most likely causal agent?
I'll give you a second to think. Just recall the age demographic and that specific gram stain morphology we talked about. Okay, ready? If you said Listeria monocytogenes, you absolutely nailed it.
Excellent. Now, I want to leave you with a final provocative thought to ponder as you continue your clinical training.
Let's hear it.
Throughout this deep dive, we've talked about how the immune system defends you and how we use antibiotics when that system is overwhelmed. But look closely at the gastrointestinal turds. The administration of broad-spectrum antibiotics can entirely wipe out a patient's normal, healthy gut flora.
The good bacteria.
Right. When you chemically remove that microscopic anatomical barrier, it clears the way for severe opportunistic infections, most notably Clostridium difficile, a spore-forming rod that causes voluminous diarrhea and devastating yellow-white colonic plaques.
Wow. So the very medicine used to save the patient opens the door for a new deadly threat, just because it wiped out the good bouncers along with the bad guys.
Precisely. So as a future clinician, how will you balance the urgent need to destroy invading pathogens with the equally critical need to preserve the patient's own native ecosystem? It is a tightrope walk that essentially defines modern infectious disease medicine.
The immune system isn't just a battlefield, it's a carefully balanced ecosystem. Sometimes the security guards at the door need a little chemical help, but you have to be incredibly careful not to burn down the nightclub in the process. Thanks for exploring the depths with us today. Keep asking why and keep connecting the dots.